I'm delighted to have the opportunity to present to you today, and thank you for this opportunity. I will give a brief overview of the therapeutic landscape for ALS, and I will do it from the perspective of an industry, as I now work with Novartis, and one of my roles is as team lead for strategy around the ALS portfolio. But at motor neuron disease, or ALS. Obviously, to all of you on the phone, very familiar to all of you. However, just a few points to note. Whilst it is an orphan disease and has orphan disease status, the lifetime risk of ALS is indeed 1 in 300. Also to note that ALS is part of a spectrum where you have pure motor disease, which is ALS, or a cognitive disorder called frontotemporal dementia. And in ALS, you can actually have both ALS and FTD. About 15 to 20% of ALS patients have progressive cognitive abnormalities. Onset can be either bulbar or limb around midlife, but we know that it can be very variable with a very variable progression, with Steve Hawken having lived almost over 50 years with ALS. Most important to note is that 10% of ALS is caused by single gene mutations that track through families. However, the remaining 90% of ALS is likely a combination of ALS and genetics. What is striking and what is really one of the challenges to note is that each patient journey is quite different, with a very variable progression. Generally speaking, spinal onset has a slower progression rate than bulbar onset, but these are generalizations, and primary lateral sclerosis and progressive muscular atrophy are slower in terms of their disease progression. We also know that if you are a carrier of a C9 mutation, you are more likely to have a faster progressing disease. And more recently, we have identified risk factors such as ANK13 that can also modify disease survival. These are important considerations as we think about clinical trials and the different progression rates in population. As I mentioned, most of ALS is likely a combination of genetics and environment. And this is illustrated here by doctors Maral Chalabi and Ola Hardeman in a review, where they show the disease as being almost a step-phased disease, where genetics carries multiple of the steps. So if you are a genetic carrier, 4 of these steps are taken up by the gene mutation. Time of cell death and cell damage is a part of what happens in the progression from birth and will influence the onset, and a small proportion, of course, environmental exposures. Once disease has begun, then it is a self-perpetuating process. So why have clinical trials for ALS been such a failure? We've had so many of them and really only 2 approved drugs, riluzole and edaravone. And so understanding this will also influence how we proceed and how we potentially have more success in the industry. I would say this is a very, very exciting opportunity for ALS. And the reality is that many more pharmaceutical companies are engaging in the disease for multiple reasons. One, of course, a very engaging ALS community globally, but also because we understand much more of the disease. And if we see from the time of identifying the disease, the progression in the last 10 to 20 years in our understanding, in the number of genes, and also, in fact, the improved symptomatic treatments that are available for patients with ALS. I think we can be very excited about the future and opportunity for finding more meaningful things. So I highlight these opportunities here, I think it's the genetics of the ALS that we can really now find novel targets, and I will highlight this a little further. A lot of investment into biomarker development. I will address why that is so important. Clinical trial design approaches, which have really changed over the years with our experience and with so many failed trials. And also the opportunity to not only look at small molecules, but approach the disease from many therapeutic angles, such as stem cell therapies, antibodies, antisense oligonucleotides, and gene therapy. And I will highlight a few of these. It's also important to note that over the years, whilst we know that motor neuron disease is really fundamentally a problem of motor neurons, these unique cells with very long axons, we also know it's not only the motor neuron that is impacted, and the surround cells and how the motor neuron connects with the muscle that is very important. And in fact, when we think of how to treat the disease, we know that the spread of the disease, is in large part due to the support cells like microglia and astrocytes. And this provides then a novel insight into which targets would be important. The genetics of ALS has been remarkable over the years. And whilst I joined in the field around 1993 with the first mutation identified for SOD1 and built some of the mouse models for ALS, it took some time for the second major breakthrough in the genetic finding for ALS to happen. However, in the last 10 years, there's been exponential increase in the number of genes found. And here, just to illustrate, SOD1 with a large circle here and C9orf with a large circle, these are still two of the largest genetic contributors to familial ALS, with 10% of ALS accounted for, 10% of familial ALS accounted for by the C9 mutations. This has really been possible through huge initiatives driven really by people with ALS, their participation, and in some cases, even their creative thinking of how to bring global teams together. And that's indicated by Project Mine, where Bernard Muller was really the founder of this concept. Answer ALS was an initiative really spearheaded and initiated by Team Gleason and Steve Gleason himself, noting how important it was for patients with the disease to really be part of the research process. And these initiatives, Create, GTAC, Answer ALS, and Project Mine, are all large repositories of clinical data, cell lines, imaging tools, really a lot of materials that can be accessed worldwide to make the discovery of treatments much more tangible. So through these efforts, we are starting to tease out how the disease occurs. We still have a way to go to understand the timing of these different things in the disease process, but we can start to put some of these gene mutations in categories, those that are involved in RNA processing, those that are involved in protein degradation, and those that are involved in cytoskeletal dynamics, the makeup of the cell itself. And we see a growing number of genes that are leading us to a clearer understanding of what are the key targets for. We see here a very complex picture, deliberately so, to say that it is a complex biology that we have to try and tease out and find drugs to make an impact on these various things. So it's unlikely that we're going to find one therapy that does it all, because there are so many mechanisms involved. But what is interesting is that multiple of the gene mutations are in multiple of the pathways. And somehow I think we are going to learn more as to what are the key nodes or the key centers that play a pivotal role, what time they come into play, and also importantly, these are clues, one, for earlier tracking of a particular disease population, and we'll talk about stratification of patients a little bit later, and also clear biomarkers for the disease. So whilst a very complicated picture, scientists have really come to a point where we can tease out and start to carefully understand and build tools for drug discovery, which has really incentivized the industry. I further wanted to note along this point where I had said that ALS is part of a spectrum with pure ALS and pure FTD. And in fact, ALS FTD carriers, or the combination of two, have genes that are common to both. And it was really the discovery of mutations in C9orf72 that consolidated the idea that there was a strong cognitive component to many of ALS cases. There are situations, in fact, the SOD mutations, which is really a pure motor disorder, and there are not cognitive features associated with it. Optoneurin is a gene found only in a pure ALS, and in frontotemporal dementia, granulin mutations are unique to frontotemporal dementia. But these findings really help us build a much better picture of the disease and of one thing to note is that TDP43, a protein found in all cell types, but really is found in abnormal accumulations in almost all cases of ALS, with the exception that SOD1 carriers and FUS carriers do not have TDP43 pathology. There are very few mutations in TDP43 linked to the disease, but we still believe that TDP43 is fundamental in some way to the biology, and we are now finding clues that C9 is linked into TDP43 biology, and risk factors like ataxin are also linked in. In frontotemporal dementia, about 45% of FTD cases are linked with TDP43 biology. The balance of FTD is tau and FUS accumulation. So what are the incentives and what are the key things that the drug development tools that the industry look for? And here I would note a huge opportunity is partnerships like those with you, with chapters that are so well connected with the community, that understand people living with the disease and have helped establish centers of excellence. This is pivotal for recruitment into clinical trials, for engaging with the community, and also learning from people with the disease how best to design clinical trials. What are the right measures? What are the outcome measures that are meaningful to people living with the disease? So a strong ALS community, which is global in fact, through the ALS Association, through the MND in the UK, through global partnerships and the International Alliance. This really builds a very, very strong network of people living with the disease, and industry really value the partnership with them. The Northeast ALS Consortium is a network of clinicians and trial sites. TRICALS, the European UK equivalent, these are all tremendous resources for the industry. The growth in academic industry partnerships, which I was pleased to be very engaged with while at the ALS Association and helped build in terms of really the first major initiatives through a partnership with industry and academia, was the Antisense opportunities at Biogen, and that has really grown from a very small initiative to many, many treatments being developed at Biogen. And other organizations such as Target ALS, who've spearheaded a very strong partnership with the industry. There is now, as I highlighted with the genetics that we've learned about, a real explosion of potential targets. Targets are already pathways or single molecules that we can think about to find drugs that intervene at these particular nodes and points. But to be successful, we really need to build up strong preclinical evidence in model systems that can be clearly matched with what is going on in the human, and really back translation, understanding our failures in clinical trials or what is happening in the population, and then going back to look at preclinical models. The development of biomarkers and also improved and better clinical trial design. With those will come a much more successful pipeline of drugs for ALS. I'm going to use C9orf72 as an example. Again, 10% of ALS is caused by these mutations. What are they? It's an expansion, a hexanucleotide expansion. We have about 30 and less in the normal population. In those with the disease, this can be in the size of hundreds or thousands. We don't quite know in detail exactly what's happening, although we've learned, and the research in this area has been quite explosive and supported by the association. We know that there are nuclear foci, and we think that this could be a pathology that is leading to the different manifestations in patients. We also know that there is protein that is generated from these expansions called DPRs. And we also know that it's possibly a combination of a gain of function with a loss of protein and a loss of function. These together all lead to the disease. What we've been able to build up, knowing that we have these mutations and these expansions, is preclinical models that have taught us a lot about what is going on in the disease, and has provided a model to try different therapeutic modalities, from flies to worms to zebrafish in mice. Whilst they don't carry all the pathologies of humans, they give us many clues. And of course, very importantly, from patients carrying these mutations, we can derive stem cell lines, which we can then further characterize in a dish. So from these models, we've been able to also get biomarkers for the disease. But biomarkers incorporate a large variety of things. So biomarkers are needed and are tools for diagnosis. We know that the earlier we can diagnose the disease, the more effective the therapies will be. Certainly for SOD1 and C9, because of the huge family histories and because of initiatives such as those by Dr. Beneter in Miami and Tim Miller, where we are able to identify patients and their families much earlier. There's huge optimism that we can actually treat earlier. Disease progression, we need markers to understand how the disease is progressing, both from a quality of life, physical strength, as well as soluble markers that can tell us quickly that we can measure in blood or in CSF. And also markers to say if we are taking drug X, does it actually engage with the target or the pathway that we thought it was supposed to be engaging with? So the kinds of markers that we could imagine are soluble, so those again that are identified either in plasma, blood, or serum, or in the cerebral spinal fluid that bathes most closely the neurons that die. We might have imaging tools, and we have had some success with building those and using them in drug discovery. Digital markers, and clinical outcome measures. Digital markers are so important. We've learned a lot through COVID, where people are not as able to get to centers, or it's not safe to travel, and even difficult to travel as the disease progresses. These are opportunities that we can measure important biomarkers whilst at one's home. And clinical outcome measures for cognition. We know that the functional rating scale, although not perfect, is at the moment the standard measure for clinical trials. And these are all under the umbrella of clinical outcome. So what are the key advances in trials? Well, again, the well-established networks, so we can easily get patients engaged in trials. We know that we have to engage multiple centers to get the right numbers of people into one trial, but these centers have established standards of operating procedures that we can be sure that if we're measuring and engaging data from one study site, it will be comparable and compatible with data coming from a different site. Also, a lot of advances in clinical trial design, staging methods, trying to understand which are the right population for the clinical trial. This is still emerging, but certainly we have already succeeded in the sense that for SOD1 treatment modalities, we only engage and enroll SOD patients. For the C9, if the target is specific to C9, we will only enroll C9 patients. And then, of course, platform trials, and I will touch very briefly on this because we'll have the opportunity to hear a lot more in an upcoming. So precision medicine is not a new concept at all. In fact, cancer is all about precision medicine, because although cancer can occur in a multitude of people, we do know that the cancers are very different and can be subdivided in populations. And so whilst a single treatment will not have benefit in the full population, we know a specific population might be a responder, and we want to try to tease out which those could be. We are starting to think about that concept in ALS as well. And this has really been very exciting, and I was privileged to be part of this in the early part of its development. And really, it's a first for the ALS field, educated in part by Alzheimer's disease, but we are now obviously showing other disease areas the benefits of this concept. And that is a platform trial. And this was really built up and led by Dr. Merit Sukovitz and her team. And it really was an engagement of multiple stakeholders: industry, FDA, patients, foundations, and investigator sites. Give a quick highlight, the real advantage of this is that we can move trials much more rapidly through the clinical trial process. We have multiple regimens going on at the same time, which can share placebos, and it reduces time, cost, and the number of placebos required. And the industry is also very much engaged with our colleagues at the Healy Center to see how they can be part of it. And in fact, the first drugs being tested are all from different biotechs and industry sector. But you'll hear a lot more about the success. In the last few minutes, I quickly want to highlight the value of having multiple therapeutic modalities. While small molecules are the easiest to potentially take, a small aspirin once, twice, or three times a day, we know that we have to be much more innovative in a challenging disease like ALS or MND. And so stem cell therapies, you've learned a lot about the beautiful work from Clive Svensson and others that have come from the preclinical studies into clinical trial settings. Antibodies, Antisense, I'll highlight quickly, Biogen's wonderful work, and then gene therapy from Avexis and Novartis, really having spearheaded the first treatment for a child. So again, importantly, different modalities also require different routes of administration. And an example here for antisense oligonucleotides, to get them to the brain, you actually have to administer either into the spaces in the brain. Here on the left, you see the ventricle, or in the spinal cord space in the lumbar spinal cord where it gets injected intrathecally. That's true for ASOs, and that is true as well for gene. And what is an ASO? Well, it's really an approach to block through a strand, an antisense strand, which then is able to block the production of a protein you don't want. And of course, we're all very familiar with the SOD1 trial that is going on. Extremely exciting, because we know that that is the cause of a small proportion of ALS. Similarly, we hope to do, and in trial is at the moment as well, the C9, which is 10% of the population. Both are going to be targeted by this approach, with C9 in trial as well, and the SOD1 trial reading out by the end. What we know from the early trial, yeah, this is Biogen's data and led by Tim Miller and others, is that lowering of the SOD protein, and we see that here, it's a dose dependent. The most important thing to note here, that with 100 milligrams, the orange line here, you significantly lower the protein that we know is causing the problems if you are a carrier of these mutations. And we also know from early data, it seems to parallel with improved ALS. Gene therapy has also been extremely exciting. It has different challenges associated with it, but we know that for SMA in 2019, it was approved, and really in a remarkable, I don't have time to show the video and the data, but for a population of children that really would no longer be able to be there. At 6 months, the severe cases would have died. There are children living beyond the age of 5 having received this gene therapy treatment. Highlight that it was actually early work from Kevin Fast and Brian Casper, funded by the ALS Association, that showed that AAV9 was an approach that could really reach these motor neurons in neonatal spinal cord. And it was this that led to the future work and that led to the approval of a now treatment for spinal muscular atrophy, motor neuron disease in children. Of course, we are hopeful that we can translate this into the ALS population as well. And this is something that many companies and academic investigators are invested in to try to find the best way to do this. And we are very hopeful with ASO and gene therapy approaches, we will have a treatment that will significantly delay and stop. So a lot of information in a short period of time, but thanks go to my current colleagues at Novartis, in particular Peggy Allred, who has been a spearhead in the ALS community, and it's a pleasure to work with her now in the industry. But my colleagues as well from Columbia, Matt Harms, I highlighted the work from GTAC very briefly, Trical's Amaral Chalabi, Niels and Healy Center, Merit Sokovitz, and really most importantly, all of you for your commitment to ALS through either donations, through those living with the disease, through your tenacity and work from an advocacy perspective. It's a real pleasure to be part of this community, and my determination continues to find a treatment that has a transformative effect on ALS. Thank you for your attention.